guys, so today we're going to do the sponge lab, and I'm just going to kind of lead you through the main things you should be looking for, but then also just follow along with your worksheet. So the simplest body plan for sponges are called asconoid. Your lab atlas calls it the Ascon body plan. It's the same thing. And the only specimen we have for that one is a slide called leucosolenia. And these sponges tend to be really little, so you can see them with your naked eye, but you're not going to see the structures until you pop it on the microscope, basically. And so what they did is these guys are really tiny and fragile and colonial, so you actually have multiple bodies on one slide, and many of them get mushed. So just try and find the nicest one. We took a picture of a pretty nice one. And so on that one, you're just supposed to label where the water is getting pulled in, which is through the top, and then it's, actually through the sides, water is getting pulled in through the sides and pushed out through the top. And again, it's a super simple body plan. For the ciconoid body plan, you actually have multiple specimens. And again, the lab atlas calls it a cicon plan. And so the main organism to look at is the grangia. That's just, again, these are all genera names, so you would italicize them or put a line under them. And you have a slide that has a long and a cross section on it. And it's stained kind of in purple. And on this slide, you will actually be able to label radial canals. So these sponges look superficially similar to the asconoid sponges, but basically their body wall is thicker and it has an increased surface area because of these radial canals. So you have a slide of grangia, but we're also going to do a dissection of grangia. They're not very big, they're about yay big, so I'm going to show you guys how to pull them out, and then you're going to use a razor blade to cut them in half, and we have photographs of what you should be looking at under the dissecting scope, both the inside and the outside of the little grangia. And we also have some bigger specimens that shows you that the ciconoid sponges can actually become quite big. The most complex of the sponges have a leuconoid or a leucon body plan, and those are the ones that, look like scrubby dub sponges. And so these tend to become the biggest sponges. And what you can label on these is these flagellated chambers. So basically water is getting drawn in through a whole bunch of openings, and is getting filtered through these chambers and shot back out through other openings. And so these have a really, really big surface area on the inside, and that's what allows them to be so big. So all of our specimens are either labeled as spongia or commercial sponge. Those are just sponges that this genus is also called the commercial sponge because it did used to be collected for people to take baths with them or used for cleaning. And so the slide is a tiny little section, and we can't call it a cross or a long section because, again, these guys are basically sort of roundish in shape. And what you'll see are little empty holes, and those are the flagellated chambers. And then you should also be able to recognize these guys in the specimen, basically in the specimen photos. And so that's pretty much it. The labs themselves are pretty straightforward, so everything in bold should be a label for a slide or a picture of a dissection or a drawing of a dissection. And then there are also other things in bold that you have to label. So if it's in bold, you somehow need to label the photos or label your drawings with those terms. Cool.